I had done some work in Peru for several years, and I had a student in my lab from Peru, Cesar, and the Montreal General Hospital one day called our lab and said, we have a Peruvian here, and I understand you have a student who's a doctor from Peru. Would you come and see this person? And so Cesar from my lab went to the hospital. He went to see this person, and they said, this person looks like he has a nasopharyngeal carcinoma, and he was starting to be treated for cancer of the nose. But he wanted to speak to a Peruvian, so my student went over there and spoke with him, and he looked in his nose. He says, he doesn't have cancer. He's got mucocutaneous leishmaniasis. And they had started to treat him for cancer, and then once he found out he had mucocutaneous leishmaniasis, he went to the tropical medicine clinic and treated it, and he was fine. So it shows how little many of the local doctors know about these kinds of diseases. But let me move to some molecular biology. This is some experiments that you can do in the laboratory. And in many ways, this sort of follows up from the talks you had yesterday from Peter, where you have like genomic information. But when you've got genomic information, what do you do then? You have to do some biological experiments. And I'll give you some example of some biological experiments that can be done in the laboratory. And I'm going to give the example of the A2 gene, which is one of the first, I'll give you one of the first genes that was really been well characterized in Leishmania, and some of the studies that were done to study a gene. So if you identify a new gene that you think is important, many of these approaches you could consider taking for some of your research. But this goes back quite a ways, in fact. When I first started working on Leishmania, and you have to have a good biological question. You have to come up with some hypothesis that you'd like to pursue. And so the question we had, and this was over 20 years, maybe 25 years ago, is that we knew that what promastigotes look like. You could grow them in the laboratory. They look like this. And then if you take promastigotes and you infect macrophages, you can see the amastigotes inside the macrophage. And the question we asked was that, what are the genes that are needed for the parasite to survive in macrophages? Those genes must be different from those genes. So there should be something genetically that allows the parasite to live in different environments. So what are those genes? So we know there's 8,000 genes. Which ones are required for survival in macrophages? Because these may be important for the pathology of the disease. So at this time, we didn't have genome sequences. We didn't have RNA-seq. So there was none of the current technologies that are available. So how do you identify a gene? Just looking at it like this. What approach could you take? Well, in fact, at that time, there was a standard approach. And what it was to make a library of all of the genes within Leishmania, and you have them in lambda-phage. And so you've got a library. All of the genes are on DNA. And then you take RNA from this stage, so genes which are expressed are in RNA, and you make radioactive RNA, converting it into cDNA. And you do the same thing here. You make RNA from these parasites, make them labeled with P32, and you hybridize that onto your library, and you look for genes that hybridize here but not here. And it was a fairly standard approach. And based on that, we came up with one gene. We called it A2 because we had a grid on a 96-well plate sort of thing, and then we had the A row and the 2 row, and that's how it became called A2. And this is a northern blot, so this is, again, we didn't have RT-PCR, so you put your total RNA on the microcellulose filter, and you hybridize it with your radioactive probe. And this was RNA from promastigotes. So this was RNA taken from this stage, put it on the gel, and you see you had no hybridization with this gene. This is macrophages here. So this is uninfected macrophages, and you had nothing here. But then when you infect the macrophages with Leishmania donovani, this gene came on. Okay, so this was the first gene that was identified that was specific to amastigotes. What was really interesting at the time is that A2, this particular clone wasn't the only one that we found. We found about 8 clones, but every one of them was A2 related. So it indicated there wasn't a lot of differences between these two, because you would expect to find a lot of different clones. But for some reason, we kept coming up with this one particular gene. And then if you look at promastigotes, again, if you just culture promastigotes and then you take amastigotes and put them in exenic amastigotes, you can see this gene really gets turned on here. So this is a really powerful expressed gene. It's specific to amastigotes. It's not present in promastigotes. So this was published back 20 years ago now. So this was the first identification of a gene like this. So I'll take you through, so what have we learned about A2 and where has it taken us in the last 20 years? And a lot of these experiments you could consider doing yourself. Well, the first thing you do, of course, is sequence the gene. We were able to do that at the time. And it had a leader sequence, which is shown here in green, meaning that it's probably directed to somewhere in the cell, or maybe even directed outside of the cell, because it's got a leader sequence. So sequences like this can tell you some interesting aspects of a gene, and Peter spoke about that yesterday. It also had this 10 amino acid sequence, which was repeated many times. There was a few differences, but it would be repeated many times. So it's not an enzyme, obviously, because it doesn't have any kind of an enzyme site. And it's got a 10 amino acid, which is repeated over and over again. So it's probably some kind of a structural protein. But maybe we can do a blast. We haven't done a blast recently, but we should probably do another blast and see if it comes up with anything. In fact, it's 10 amino acids, but it's really 5 that's repeated twice, because you can see VGPLS and then VGPQS. So it's almost a repeat of a repeat. So this kind of sequence doesn't tell you a lot about what it may do, but we can still do experiments to try and understand what its function is. So we also looked at the genes along the chromosome, and we know that there are 2 chromosomes. And the A2 genes are shown here in white. So the first thing we found out is that there are many of these genes. They're not just a single gene, there are many copies of it. So it's a multi-gene family. And you have A2 genes here, which are shown in the white blocks. And you can see that some of these genes, some of these white blocks are bigger than other white blocks. And the reason is because these ones have more repeats than these ones. So that 10 amino acid repeat, some of them have many more repeats than others. So this would be a big one, so it would express a bigger protein. This would be a small one, it would express a smaller protein. So there are many genes expressing A2 of many different sizes in it. And if you look at a Western blot, so we produced antibodies against this protein. We made very good monoclonal antibodies, and the monoclonal antibodies recognize one of those 10 amino acid repeats. So these are really excellent antibodies to study A2. And on the Western blot, you can see promastigotes express very low levels of these A2 proteins, but in the amastigotes, you get many bands. And these are the same protein, it's just different number of repeats coming from the different genes. So you have here one of high molecular weight, so it's got many repeats, and low molecular weight, so it has fewer repeats. Another thing you can ask when you're trying to understand the function of a protein is, where is it within the cell? Because sometimes that gives you some indication of what function it may have. We haven't really pinned this down completely, but this is an amastigote where we've done using our antibodies, immunofluorescence. And if you stain with A2, you see this is within the amastigote. You get some bright colors here, indicating this is where the A2 proteins are. But that doesn't tell you much, just because you have spots within the cell. You have to sort of compare it to another protein that's already been identified. And so we compared it to a number of other proteins. One of them was BIP. BIP is a protein which is specific to the endoplasmic reticulum. In fact, what it does is it takes proteins and helps them become incorporated into the endoplasmic reticulum. And if you look at the staining pattern of BIP and the staining pattern of A2, you can see it's very similar, and they overlap here, indicating that at least some of A2, it looks like it's getting into the endoplasmic reticulum. So when you think about the function of the protein now, well, it's something that may have to work in the ER, or the endoplasmic reticulum. So that's another clue that you could use to see what the function of A2 might be. And there was another interesting aspect of A2 was that if you infect macrophages, and this is a macrophage cell here, the nucleus of the macrophage cell, and this is heavily infected with amastigotes. So these are amastigotes shown here. And you can, and the green is where the A2 proteins are expressed. And one of the things that's very obvious is that not all of the amastigotes are expressing A2. Some of them express high levels, as shown here. Some of them express very low levels, and some of them appear not to express any at all. So it's not a constitutively turned on gene. We know it gets turned on in amastigotes, but it's not all of them that are expressing the same amount at the same time. There seems to be some order or some other reason for expressing A2 in addition to just being an amastigote, because these are true amastigotes, but they're not all expressing the same amount of A2. Does that help us understand the function? Maybe, I don't know. I mean, it might help you think about some things. And I'll give you, we'll come to a reason why maybe this may be happening. The other thing you can ask is, well, all of this is done in the laboratory, but is A2 actually really expressed in the field? You know, when someone is infected with Leishmania donovani, are these people really, is the protein actually really there? So one way you can do that is see if people make antibodies against A2. If they make antibodies, that means the A2 was there. So this was one experiment done by Elodie, who was a graduate student in my lab. And she took patients from VL, at this time we had some sera from the Sudan, and many of these, almost over 90% of them, had antibodies against A2. So clearly this protein is being expressed. And this also indicates it might be a useful diagnostic antigen as well. So this was one of the things that we considered using A2. It's not being used as a diagnostic antigen, but at least this experiment shows that it is present in people. So one of the other important tools we have in Leishmania is you can knock genes out. So you can grow the parasite, and genetically you could remove genes from the genome. And this is one of the most powerful techniques that we have in Leishmania. I don't have time to go through the technique. I'll be happy to discuss it with people, but it's basically you define the flanking regions of the gene, and you put in a antibiotic-resistant gene, and through homologous recombination, you can replace your gene with an antibiotic-resistant gene. This was difficult to do with A2 because there are many genes, so it's impossible to get rid of all of them this way. But we were able to get rid of some of them. So this is a southern blot, which shows, these are the DNA, which shows the different genes present in Leishmania donovani. And through this technique, we were able to remove this gene. So you see this gene is present in the wild type. We could remove it from this type of parasite. And when we removed it, we removed the protein. See, this was a big gene, and you remove the protein here in the western blot. Here we were able to remove several genes, this one, and one here. So we were able to reduce the number of genes in this one by homologous recombination. And you can see we reduced the number of proteins here as well. So you can knock out genes. It's much easier to do this with a single copy gene, obviously, because you don't have all of these genes to get rid of. But if you knock out the genes, you then can ask a question, does it change the biology of the parasite? And one of the easiest things you can do is inject it into a mouse and see what happens. So if you do this experiment and you inject these into a mouse, what you find is this is your wild type parasite here. So this is the one that has all of the A2. And this is the type 2 where you remove one copy, type 1 where you remove several copies. As you start to remove A2, you remove the ability of Leishmania to survive in the liver. So this is an indication that these genes are expressed in amastigotes. They also appear to be needed for it to survive, because as you start to remove them, you start to remove the ability of the parasite to survive in the mouse. So this is a really powerful approach that we have in studying Leishmania genes. And this is, makes Leishmania a very useful parasite to study at this level, because we can do these kinds of genetic changes. I won't talk about those ones. I'll just keep on going. The other thing you should do when you identify a gene that you think is different or you think is interesting is you can ask, well, is it present in all species of Leishmania, or is it present in only some species of Leishmania? And this also could help you identify the function. So this, again, was an experiment that we did And in this case, it wasn't a typical southern blot. It was a chromosomal, looking at chromosomes and where A2 was. And so we looked at several parasites to see where A2 was. And you can see that these are Donovanis here, Leishmania donovani, and you can see the gene is present in Leishmania donovani. It was present in Leishmania mexicana, so that was clear from here. But what was interesting, the gene wasn't present in Leishmania tropica or Leishmania major. So this is a gene which isn't present in all species. So this may also help us understand its function. So the question is, these are old world leishmaniasis which don't have the gene, and donovani is also an old world leishmaniasis, and it does have the gene. So some genes are not present in all species, and this was one of the ones. So if it's not present, so we looked in L major to find out why the gene isn't present there. And there was something that this was really very interesting when we compared the different leishmanias. And so this is the Leishmania donovani gene here. It's got the open reading frame, which has many repeats, and it has a 5' untranslated region, a 3' untranslated region. When we looked in the Leishmania major, we found that the coding part of the gene was completely collapsed, completely reduced. Many of those coding regions were gone. So if you look at the protein, you had each one of these boxes that 10 amino acid repeat, so you had all of these repeats in donovani, but when you looked in L major, there was only one single repeat left. But what was really interesting is it still had the same leader sequence and the extreme 3' end. So, which argues that L major at one time had the gene, but for some reason, over evolution, it just kept getting smaller and smaller and smaller, and it got rid of the gene. So why would L major not want this gene anymore? So at some reason, L major had it, but it lost it. So that's also a clue as to maybe why. And obviously, these parasites are quite similar, but this one causes visceral disease, where the parasite goes inside the body. This one causes skin disease, where the parasite stays in the skin. And this one has lost the A2 gene. So the next thing you can do is add back a gene. So the experiment we did then was what happens if you take this gene and put it back into L major. So L major lost the gene in evolution. Let's put it back and see what it does. And so this you can do again by a transfection. This is a A2 plasmid. It's normally circular, but I'm drawing it in a linear form here. So we have the one copy of the A2 gene, the 5' end, the 3' end. And then we have this control vector. This is a vector that we in my lab made. So you can put any gene into this vector. We have a vector. So if you want to express a gene in Leishmania, you can write to me. I'll send you this plasmid, and you could replace A2 with any gene you want to express in Leishmania. And then we transfected this plasmid into Leishmania major, and you can see what was quite interesting is that it's still highly regulated, even though it's expressed from this plasmid. In the amastigotes, you had high levels of protein, but when you culture a promastigote form, the protein wasn't there, even though it had the plasmid. But in the promastigote, for some reason, the promastigote doesn't like to make this protein, or it's not normally expressed in promastigotes. But when you switch it to the amastigote, it's expressed, even though it's coming from a plasmid. It's not coming from the chromosome. So that was quite interesting. But the system worked very well. So did we change the biology of the parasite by putting in this single gene? And you do intravenous infection in the mouse, and then we looked in the liver and spleen, and yes, we found that this would be the spleen of a control mouse, which has the plasmid without A2, and if you had A2 in there, you see you had a larger spleen, and you had higher levels of infection as well. So just by genetically changing one gene, you can convert from a parasite to become more virulent and help its ability to survive inside the liver and spleen. And this is, of course, what you see in a visceral leishmaniasis, where one of the common features is splenomegalia. And you can see the A2 here induces a splenomegalia. So this is a gene that probably is involved with this visceralization and induction of the splenomegalia. So what is A2 doing? apart from allowing the parasite to survive in the spleen, how does it do that? Well, one of the other things we looked at is what also induces A2 expression. And what we find is that if you just take your Leishmania donovani and you heat it to 40 degrees, these are A2 proteins are induced here. So this is if you grow Leishmania at 26 degrees, you have very low levels of expression. But if you heat them to 40 degrees, the A2 proteins come on. So this isn't differentiation from promastigotes to amastigotes. This is just heating the promastigotes. So you can actually induce the parasite by heating at 40 degrees, as shown here. You could also induce it by other kinds of stress. And here we use ways of unfolding the protein. So DDT causes protein unfolding. It induces A2. Tunicomycin causes alterations in the protein, and you can get some induction. So it seems to be induced by some stress. So what we did, we were able to knock down the A2, and this was through another approach. But if you remove A2 from Leishmania donovani, what we find is at 40 degrees for 12 hours, the parasite survives quite well. But if you take away A2, the parasite doesn't survive as well. So it needs A2 to survive at a higher temperature. And this is quite an interesting experiment as well. And again, it involves looking at survival in different heat. So L major, which stays on the skin. In this assay, if L major is grown in these wells here, and if the parasite is alive, the color of the LMR blue is red. So if the parasite's alive, it's red. If the parasite dies, the color of the dye goes blue. So if you take L major, grow it at 26 degrees, it's alive. It's red. If you switch that culture to 40 degrees, L major turns blue, means it's dead. So L major cannot survive at a high temperature. It has to survive at a low temperature, 26 degrees. If you compare to donovani, at 26 degrees, of course, it's alive. You switch it to 40 degrees, it stays alive. So donovani can live at high temperature much better than Leishmania major. If we take the A2 gene and put it into L major, you see you get some intermediate here. Just by moving that gene in there, it allows the parasite to survive better at a higher temperature. So at this point, our working hypothesis is that A2 is needed for the parasite to survive in the visceral organs. Particularly, one of the common features of Leishmania donovani is that you have fever. So when you have a fever, fever is there to try and kill any kind of pathogen we have. But even with fever, A2 somehow helps to protect the donovani from dying. And if it's not there, then the parasite has to stay on the skin because it won't survive inside the body. And so
PKDL parasite will not express it?
There has been some study. In fact, there has been some study, and the PKDL on the, if you take the PKDL, I believe there is less A2 on those PKDL than internally. Soltra published a paper on that. But that's a good question. So these are some studies we were doing in the laboratory, and then several years ago, I got a call from somebody in Sri Lanka. This is when I was still working at the WHO, and her name was Shrilinda Ranasinghe, and she said, you know, we have a problem of cutaneous leishmaniasis in Sri Lanka, and I'd like some help to try and do some PCR and identify which kind of parasite it is. So she contacted me, and she said, because there's evidence that this parasite is Leishmania donovani, but it's causing cutaneous leishmaniasis. Of course, everywhere in the world, donovani causes visceral leishmaniasis. So I said, well, that's very interesting. Will you send me some of that parasite? We'd like to look at it. So she sent the parasite from Sri Lanka to my laboratory in Montreal, and we started to look at it. And I'll talk a little bit about that now. But this is the kind of lesion that this causes, and it's a Leishmania donovani. You don't see this anywhere else, Leishmania donovani causing skin lesion. Leishmania donovani always gets inside the body. It goes inside the liver and spleen. So we were very interested in this parasite. The other thing that's interesting, so this is a part of Sri Lanka that had Leishmania donovani causing cutaneous leishmaniasis. And that was all of these red dots here. This is where somebody was living. They had a cutaneous leishmaniasis. And there are, in fact, thousands of cases of cutaneous leishmaniasis in Sri Lanka, all caused by donovani. But in the midst of all these people, there was one person here who had visceral leishmaniasis. So even though you have a lot of cutaneous leishmaniasis, there was one with visceral leishmaniasis. So Shrilinda was really terrific, and she was able to get parasite from this person as well as parasite from a cutaneous. So not only did we have the cutaneous parasite, but we could compare it to a visceral parasite as well. So these samples were incredibly valuable, particularly this visceral form, because there's so few of these. So we got these parasites. She sent them from Sri Lanka. In fact, you can send, you have to have the proper papers and authority to send parasites. But in fact, you can send live parasites, Leishmania promassicles. They survive quite well. You don't have to freeze them down, put them in liquid nitrogen, and then try to send them, which is very difficult. If you had a culture of parasite, you can send them from one part of the world to the other. They'll probably survive. You could take it out and start to culture it. So we actually sent her the media because she didn't have the media to grow the parasite. It got there. She took the parasite directly from the patient, one from the nose, another one from the bone marrow, and put it into the vials that we sent, grew them for a few days, and then sent them to Montreal just by regular post. And they actually got there alive, so they went halfway around the world. So now you've got two vials. Okay, you've got a cutaneous leishmaniasis vial, one from a cutaneous patient, and you've got one from a visceral patient. What would you do with them? It's a question. This is a quiz question like Peter was asking you yesterday. What would you do with them? You have your laboratory now. You've got these two cultures.
Something regarding the protein that you were talking about?
Maybe.
See if it's expressed in this.
Yeah. What would you do before that, though? Okay, let me give you a. Okay.
First one you have to grow and preserve its parasite.
Okay. Yes. But what would be? Okay. Let me ask you this question: what would Koch have done? You know, the Koch postulates for looking at determining whether a pathogen causes a disease. What would Koch have done 100 years ago? He didn't have any tools. He didn't have any of the kind of tools that we have today. Pardon? Right, that's what Koch would have done. So that's what we did first. We put them into a mouse to see what kind of disease they would cause in a mouse. Very simple experiment. I think it's here. No, that's not here. So what we did is we got this is the patient that we got the parasite from. This is the patient that we got the cutaneous lesion from. This is what the parasite looks under culture, exactly the same. And the theory at the time was it's just the same parasite. Some people get visceral disease from this parasite, and some people get cutaneous disease from this parasite. So that was the theory at the time. But we got the parasites from both of these individuals, and we injected it into the mouse. And so when you inject it into the mouse, in Leishmania donovani doesn't go from the skin to the liver and spleen, so it's not a perfect model. What you have to do is inject it in the tail vein, and then 4 weeks later look inside the liver and spleen to see if it's surviving in the liver and spleen. So the result we got was really, really interesting, and it wasn't one that I was expecting. But the cutaneous parasite was not able to survive in the liver and spleen. You had very, very low levels of parasite here. Whereas the one from the visceral patient was able to survive. It was very virulent within the liver and spleen. So it had changed its pathology. These were different parasites. They were both donovani, but one was able to survive in the liver and spleen, the other one wasn't. So there was a switch. And that's why this one somehow now has developed to live in the nose, because it can't live in the liver and spleen. It doesn't survive in the liver and spleen. So at this point, and in fact this was at one of these meetings, we had just gotten this result, and I was talking to Peter, and obviously there was an obvious thing is that we should sequence these now that they've got different phenotypes. So Peter talked a little bit about this yesterday, so I'm not going to go into detail about all of the sequence information because you got enough of that yesterday. But what did we find when we sequenced the genome? So first of all, Leishmania genome is about 32 megabytes, 8,000 genes. what were the major observations from sequencing? And you can go back, the paper recently came out in PLOS, so you could look at all of the details, which I'm not going to give you here. But the thing that I found, the major things that I found very interesting is that the two Sri Lankan strains, the cutaneous strain and the visceral strain, were very closely related. In fact, these two Sri Lankan strains, even though they cause different diseases, they were more similar to each other than the Leishmania donovani from India. India, if you compare to the Indian strain, these donovanis were quite different. So it's a strain of donovani specific to Sri Lanka. It's diverged into a cutaneous and a visceral, but they're still genetically very closely related. The other thing I expected is that there would be some genes that were lost or genes that were gained. There would be some major genetic changes. In fact, we didn't find any real evidence for pseudogenes or genes lost. So they had pretty much the same genes in the two strains. There wasn't any obvious thing that was gone, which we found surprising. So the only thing so then you have to start looking at other things. And the only thing we found was that there was some non-synonymous SNPs. So this is a SNP where you've changed the amino acid within a protein. So that, we did find quite a lot of evidence for that. And we found that in one gene called A2. The other thing, of course, we would look at A2. I mean, this is the gene that we've been looking at for a long time, and what's happening with A2. And what we did find is that there was differences in the A2 genes as well. But let me just talk about this RAG-C for a minute. Also, yesterday, you heard a lot about pathways and looking at how metabolic pathways could be important. Well, this is one of the most important pathways in eukaryotic cells. It's called the Mtor pathway, which is an important regulatory pathway. In higher eukaryotes, this pathway responds to nutrients. So nutrients induce RAG-C activation, reduces Mtor, and it reduces proliferation. So this is the common pathway in eukaryotes. And this is the M, here is called M, the M isn't here, but it's the Tor here. And Leishmania actually has 3 of these genes. So this pathway appears to be present in Leishmania. But those genes were all normal. What was interesting is that we had an interesting polymorphism in this RAG-C gene here, which is an important regulatory of this one. So when you're looking at SNPs, so we had SNPs in many of the genes. We didn't know which ones are important. You look for things that are a change in the amino acid where it seems to be an amino acid which is well conserved or within the active site of a gene. And so we had a number of SNPs that are all shown here. The SNP in this RAG-C was in this gene here on chromosome 36. And you could see in the wild type L. donovani, it was arginine. It was an arginine in the visceral. And it switched to a cysteine in the cutaneous form, and everywhere else it was an arginine. So this is one way of maybe looking for important SNPs. And so these were a number of other genes that had SNPs that we thought were interesting. And we transfected the normal copy of these genes back into CL, put them back into the mouse, and the only one that increased infection levels was this RAG-C gene here. So by converting this C back to an R, we could go from a low level of infection to a high level of infection. So this is it just shows you how remarkable the genome is, because in one gene, you change one amino acid, you can change the pathology, you can change the phenotype of the parasite. So this is why you don't have to have gene deletions. All you have to have is changes in a single amino acid. And you can convert this into a more virulent parasite, as shown here in the levels in the spleen. So that was one of the changes that we think is important for this change in phenotype. The other one was the A2 gene, and Peter showed you some of his results yesterday. When you sequence across the genome, and this in the visceral form, these are the red sequences here, and the cutaneous form is in the green sequences. You have more red sequences, meaning you have more A2 genes here. And when you do the Western blots to look at proteins, you can see in the visceral form, you have more A2 proteins here than here. So somehow, it's almost like with L major. Remember L major, I told you it started to lose the A2 genes until it got nothing? Well, these parasites seem to be undergoing that same process where you had the visceral form, which has more A2 proteins, but it seems to be reducing its A2. And then it's to the point now where it's no longer behaving like a visceral parasite and it's behaving more like a cutaneous parasite. Again, you can do the experiment of adding it back. So we add one more copy of A2 back into the cutaneous form, and we could increase the level of infection. So there has to be a threshold level of A2 for the parasite to survive in the liver and spleen. So I think, so we have a number of ongoing studies. And these are questions we still don't know what's going on. Why has the CL form lost its virulence? Why did it switch from a visceral parasite to a cutaneous parasite? And one of the things that I think we should be looking at, and we're starting to look at, maybe humans are no longer the reservoir for this parasite. Maybe it's moved from a human reservoir back into an animal reservoir. And when you move to an animal reservoir, it may not need to survive at the higher temperature. So it might be one of a Donovani that has switched to an animal reservoir. And we have some evidence based on, well, I won't go into that, but we have some weak evidence, and we'd like to follow up on that. Another question is, why are there so few cases of visceral leishmaniasis? The parasite is there, but why are there so few cases? In fact, you have some years there's not even a single case, but you have so many cases of cutaneous leishmaniasis. Why? Why is that? The parasite is there. You would expect to have some cases. So one reason might be is because if you have a lot of people getting the cutaneous leishmaniasis form, they may be immunized against the visceral form. So people get the cutaneous form, and now their immune system is protecting them from the visceral form. And we have some evidence in the mouse that that's the case, whereas if we infect with the cutaneous form, they are protected against the visceral form. And this was recently published in Vaccine. Again, you know, what are the function of A2 and RAG-C? This is something that needs to be looked at more at the molecular level. But the other problem we have is we have thousands of SNPs, basically. How do you know which ones are important, which ones aren't? Okay, so we still have that problem. We can identify some of them manually, and we think some of them might be important, but we're still guessing at this point. So how do you address that problem? So one way we're starting to look at that is we've taken this cutaneous parasite form, which normally doesn't survive in the spleen of mice, but we're trying to force it to be able to survive in the spleen. So we're trying to convert it from a cutaneous parasite back into a visceral parasite. So how do you do that? Well, this is some preliminary data now. And this is infection in the spleen. So if we take the cutaneous parasite, we injected it into the tail vein. 1 month later, we go into the spleen, and there's very little parasite. But there is some. After a month, some of the parasites do survive. So we take these parasites, grow them up, and put them back into the mouse again, and allow it to go for 1 month. So after a month, we take the parasite out. There's a little bit there. We grow it up. We put it back into the mouse again. We wait a month, take it back out of the spleen. And this is the level of infection. And we keep doing that. We did this for 5 months. When we got to 6 months, all of a sudden we got a parasite that could survive better in the spleen. So by forcing it to survive in the spleen, it's starting to survive in the spleen. 7 months, we got a higher level here. So we're starting to move the parasite into a higher level of infection by forcing it to survive in the spleen. 8 months, we went down here. What do you think happened there? We had a contamination. We got the parasite out of the spleen, and it was contaminated with, it was a yeast contamination, which can happen by doing this. So we thought we had lost this culture. It was so precious to us, and it became contaminated, and we said, oh, you know, what can we do with this culture now? It's contaminated. But what we did is we put it back into the mouse anyway. Even though we weren't able to grow it up, we just immediately put it back into the mouse. We waited a month, and of course, and we were, you know, there were still some here. We were able to pull this parasite out. Some of them did survive. We grew it up, put it back in the spleen again, and it resumed to have a high level of virulence now. So we were able to recover it, and it still has a higher level of virulence. So now we're in the process of sequencing this one and compare the SNPs from this parasite, the original, to this one that we've had for now a year passage through mouse. And this may be another way of looking at what are the most relevant types of changes. So in summary for A2, what I can say, or our model is the following. A2 allows the parasite to live in fever. It also provides some protection against reactive oxygen species and nitric oxygen species. I haven't shown you those data, but we have those data. So basically, A2 allows the parasite to survive in visceral organs. If it's gone, it doesn't do very well in visceral organs. So L major doesn't have A2, so it's got decreased ability to survive in fever, decreased ability to survive in internal organs. If it can't do this, it's got to survive somewhere, so it develops a way to survive in cutaneous leishmaniasis. So what are some of the applications for A2? Well, people like to use it as a differentiation marker when you grow your amastigotes exenically and you want to say they're exenic amastigotes, you should see A2. So it's a differentiation marker. It tells you you've got a difference between promastigotes and amastigotes. You can use it to develop a plasmid to express other genes, and we have the A2 3' sequence and 5' sequence, and we use this as our backbone plasmid to express genes in Leishmania. Potential to use it has a diagnostic test because most people produce antibodies against it. And what about a vaccine? Can this thing be used as a vaccine? And this is not work that's been done in my lab, but there's a group in Brazil who's been working and doing a lot of work looking at the use of A2 as a vaccine. And A2 is now the licensed. It's gone through phase 3 clinical trials in dogs. The trial involved 1,600 dogs and showed that it was protective in dogs. And so there is now a commercial product called Leishtec, and it's a canine vaccine which is now used. It's one of the 2 licensed vaccines present in Brazil. And Ricardo has also looked in the monkey model, looking at macaques, where he's vaccinated macaques using adenovirus expressing A2, and he found that he's got very good protection in macaques as well. So not only are these really interesting molecules to study in the laboratory, but some of them may also be useful as vaccines. So I'm not saying that because it's got that biological activity, it's a good vaccine, but it may be, as a key molecule, it might have ability to also provide some direction for vaccine development. And A2 is also one of the candidate vaccines that people are now considering. So with that, I'll be happy to answer questions. Thank you.